Hello everybody, and welcome. I'm Tess Harris of the PKD charity, and it's my pleasure to introduce you to Dr. Evi Goggolidou, who's a reader in molecular genetics at the University of Wolverhampton, who's going to give us a research update into autosomal recessive polycystic kidney disease, or ARPKD. Thank you very much for joining us, Evi.
Thank you for the kind invitation and your really kind words. So today we're going to concentrate on some recent findings we have on ARPKD, and I will try and give you an overview of what we do in the lab, but also discuss a couple of points that are very current. So we know that there's two forms of PKD, the dominant, which usually appears later on in life, and the recessive that I'm going to talk about today. Both are inherited. Some of the words here are from our genetics lecture, so don't worry so much about the terminology. I'll explain everything. What is interesting between the recessive form, ARPKD, and the dominant is the genes that cause the disease. So both dominant and recessive PKD have links to cilia, and it is, I don't know how, this is not taking me to the next slide. Sorry about that. Okay, that's better. But they both have links to cilia, and cilia are the antenna that our cell uses to either transmit information within the cell or to neighboring cells so that organs can function. Sometimes those cilia move, but in the case of the kidney that is affected in PKD, we're looking at cilia as an antenna. So a structure that forms from the cell itself at a specific time point. It does perform certain functions in terms of signaling, basically. So there is a need for an action to be taken. The cilium can facilitate this action. And then when that cannot happen, we could potentially end up with a number of diseases, one of which is PKD. And there's a particular pathway that we work on in the lab, which is called WNT signaling. Don't worry about the various names of the proteins. The proteins are the building blocks of the cell. So this is what we need to have a fully functional cell. And in the case of this pathway, what I have done here is I've listed a number of players. So this is not all the players for this particular pathway. These are just the key elements of the pathway. What we have shown in the past, if you look at the right-hand side of the picture, is that we have got, when we have some of these proteins, either not positioning themselves in the right place in the cell, and this serves, so the position of these building blocks on the cell can serve as a signal, as a kind of indication to the cell itself or to other proteins to conduct downstream functions, to perform functions. So when they cannot find a protein at a specific place, the cell can get confused perhaps, or not be 100% clear as to what needs to happen. And in the context of the pathway on the right-hand side, the confusion in terms of cellular behavior in very simple terms is that it doesn't realize the cell itself cannot detect its neighboring cells on the plane, or it doesn't position itself right. So the organ itself cannot form. This also has effects in transcription, and transcription is the process between the DNA and the proteins. The proteins are the building blocks. So if the in-between process is not narrated correctly, then the building blocks cannot exist. What we have found in the past is that many of these have some links with PKD. Others have shown links with ADPKD. We have shown links with ARPKD. And this is where our involvement with ARPKD started. So what has been shown is that many of them are altered in PKD, and on the left-hand side, this applies both to AD, to dominant and recessive. So on the right-hand side, you can see a normal kidney, and you can see some names which are presence or expression of these proteins. So they are either forming or they perform a function. And on the right-hand side, you can see a polycystic kidney, and you can see many names I'm listing which are different. This can be that there's more of them, or there's less of them, or they sit in the wrong place, or they don't form right, so they cannot perform the function. You know this way better than me. From a scientific perspective, all I'm going to say is that ARPKD has manifestations. It can have manifestations only in the kidney, only in the liver, or both in the kidney and the liver. It has a low, well, a rare incidence. It's a rare disease, so it has a low incidence, 1 in 20,000. It manifests equally in boys and girls, and depending on the age of manifestation, the mortality rate can be different, but it appears that the critical window is in the first 1 or couple of years of life. And what we have decided to do is, what I'm showing you here is a study from 2003. So I'm sure there's more recent data that's been collected. But this got us thinking on why there is such range in severity. Why is it that some children with ARPKD are absolutely fine until a much later age, whereas others are diagnosed at birth and they don't have a very positive outcome? Why is it that some kids have manifestation predominantly in the liver, while others have manifestations predominantly in the kidney? And this is so I'm a geneticist by training. From a genetics perspective, I found that very interesting, because what we do know is that, don't worry about this, this is way too much information. What we do know is that there's 2 genes that are relevant to ARPKD, PKHD1, that when has mutations, so when there's changes in that gene, ARPKD manifests for the majority of the cases. So most children with ARPKD have changes in PKHD1. There is a percentage, a smaller percentage, that has changes in another gene called DZP1L, and both of these genes have links to cilia. So either they are placed on cilia at some point in their life, or in the case of the later gene, they also are responsible for cilia formation. And this is why Tess mentioned before, this is one of the reasons why the ciliopathies and the cilia alliance is very important for PKD. I think I've shown this before, but what our first question in terms of understanding the range of severity was to look at whether in that population of patients that we had, thanks to the PKD charity and the biobank that exists at UCL, whether they had changes. When I say mutations, I mean changes, so the term is intervariable, in only PKHD1 or in PKHD1 and DZP1L, and where exactly those changes were found. It's important to say here that PKHD1 is a very big gene. So the size of the gene determines the difficulty of the process in terms of getting those building blocks to form normally and work. And what we did see, and is summarized here, so we had a group of patients, different severities of disease, and we made the hypothesis that more severe patients perhaps would have changes in the DNA in a specific part of the gene, while others would with a milder manifestation. So older children that reached end-stage renal disease, that lost kidney function at a later point, would have changes in DNA in that particular gene at different positions. And we tried to find hotspots. So we thought, I think I have a schematic here. Yeah. So if you look at the bottom, that's well, I guess we're being artistic. That's our presentation of the protein, which when it doesn't function right, it gives rise to ARPKD. And you can see that this protein has got specific domains. So each of these domains are the parts of it that are giving the signal for it to perform a certain function. We assumed that we're going to find certain signals, certain parts of it that affect the downstream function affected in severe and others in moderate or mild, but we didn't see that. So for our cohort, which is exclusively patients with mutations in PKHD1, we didn't have any patients with mutations in DZP1L. We found that actually the position of the mutation doesn't affect the outcome. So you can have individuals that reach end-stage kidney disease in the first year of life, and individuals that don't reach end-stage renal disease until 15 or 18 years of age that have mutations in similar places or similar types of mutations. So it's not actually the information itself in the context of the gene that appears to be affected in this cohort. There is something else. But we didn't know what this something else was. So theories that perhaps you might be interested in or already be familiar with is that there is this, so again, from a genetics perspective, you might have a change in a gene, but that change is not as severe because we all have about 18,000 genes, and some of them might have a protective role. So if you have a change in gene A, but you have this gene B that has a very strong expression, this could mask the severity of the outcome. Or the opposite, you might have a gene C that has a certain sequence that causes a more severe outcome. It's very difficult, though, to study relationships between genes and how one affects the other. We are currently doing that, and that's hopefully going to be, when we are invited to do a research update, I can tell you more about these relationships. But a kind of initial way of us to understand why is it that there isn't a hotspot for mutations was to look at what happens downstream. So what happens, we have the DNA, we've looked at it, and it appears that the information on the DNA doesn't affect the outcome. But if you remember from my previous slide, then we also have the building blocks, which are the proteins, which are downstream. So could it be that actually what we have is that the change in the DNA is affecting the building blocks, many building blocks that the cells need to function? And to answer this, we decided to look at mRNA. So I think since COVID, we're all quite familiar with mRNA because of the vaccines, and we know that mRNA is the signal that makes those proteins. So we looked at signals from a range of patients, and we compared them to normal kidneys. When I say patients, we actually looked at kidneys taken at nephrectomy, and we compared them to normal kidneys to try and understand whether any of the building blocks are different. The names you see there are examples of, so we did find differences, and the names you see there are examples of these, of some of these building blocks, one of which was Wnt9b. And if you remember, we happened to be working on that signaling pathway. So this further indicated to us that it is an important pathway for ARPKD. These names are not part, I mean, I can tell you more about them if you want me to, or I'm happy to answer questions about them. But what this shows is that there's a clear difference in the expression of certain building blocks, messages that lead to building blocks that are different between normal kidneys and ARPKD kidneys. And you might ask, okay, so you have names of genes there. How is this gonna make a difference? What we did do is we tried to dissect this further, because once we get a name of maybe a list of genes or a specific gene, what we can do is look at approaches to restore them to normal levels. So if we look at, let's look at the left-hand side, anything that is marked in red and has a name next to it has greater expression, so there's more of it in ARPKD. And that to me, that says that if in normal situations there's less, and in ARPKD there's more, there could be a link between this higher expression and the disease itself or its severity. So there's many ways you can go about it. One is, can we try and understand this better and potentially look at bringing it down to normal and see if that slows down the disease? Or you might want to see whether this is different between the different severities. Is it different in children that have both a kidney and a liver manifestation? Because here we only looked at the kidney, so I cannot tell you anything about the liver manifestation yet, although we have a project planned for the future. So there's many kind of questions and areas that this is bringing together. And also the other approach we can take is, instead of looking at names of genes that we can directly try and manipulate, either increase or decrease or change the expression of or the localization of, is to look at pathways collectively. So is there a specific, by pathways I mean specific processes that the cell normally performs that in ARPKD performs slightly differently? Which are they? How are they relevant? What do we understand by them? So if you look at the left-hand side again, where I have the gene count diagram, you can see that metabolism, cellular metabolism, I mean, comes up quite high, as do processes related to the cytoskeleton. So the cytoskeleton is the scaffold of the cell that keeps everything in place. And the fact that, and it's very relevant to cilia, because cilia sit on the cytoskeleton to form. So the fact that we have detected changes both in metabolism and the cytoskeleton tell us that in ARPKD, the cell morphology and function is severely affected, which is, I mean, I suppose it's not particularly, it's kind of expected, because you can see that the kidneys have lots of cysts, they're not functioning fine, they're increased in size, so it makes sense that the cells themselves are affected. But here what we have is particular processes that are affected. So we can investigate them further and try and see how we can bring them back to normal. And maybe I can say this and then stop and take questions. What we do have as well, because it's very complicated to study the liver element of ARPKD and the kidney element in individuals at the same time. The work that I presented on the kidney has taken us 4 years, and we're very grateful to the PKD charity for the funding. So to look at the kidney and the liver together and try and get a better understanding, we have an animal model that appears to have both the kidney and the liver affected. And what we're doing there is we're trying to see how this process arises and whether any of the changes in Wnt signaling that I mentioned before are relevant, and how we can, if we were to start manipulating them, how we can perhaps slow down or restore to normal the kidney and the liver changes that we see in ARPKD. So I think that's enough from me. I'm happy to take questions from you. Anything related to names or anything I've said perhaps that doesn't make much sense, I'm happy to answer.
Thank you very much, Evi. So do we want to leave the slides on, or shall we take the slides down? And is everybody happy to show themselves, or would you prefer that I stop recording now at this stage?
I'm happy for, so the slides can stay as they are for now. Mm-hmm. I can take questions, and people can show their face, or they can type in the chat box.
Okay. Is that if everybody's all right?
Okay,
You can see the chat box, so you can use
anything.
that if you don't wish to show yourself at this stage. But as Evi said, do feel free to ask questions. I know that was, thanks very much. That was interesting, but obviously a very scientific presentation. Karen, you have a question.
I don't actually have a question. I just want to say thanks, Evi. That was really helpful. I thought I would just put my camera on just so that you could, didn't feel it was you that had your camera on, Evi. But thank you. I really appreciate everything, all the hard work that you're doing, and everything that's gone into the presentation today. So thank you.
Thank you so much, Karen. That's really positive to hear. And as Tess said, it is a very scientific presentation that we're currently putting together to send out to peer review papers. So I didn't want to tone it down, because sure, there's a couple of terms there that maybe they don't make immediate sense. It's important. Those terms, however, are important in terms of the scientific element of the disease, and it does have a very strong scientific element. We know there's 2 genes involved. The problem we keep having all the time, it's a problem for us as scientists. You all have separate problems, which I completely appreciate, and they're way bigger than mine. But the problem we have as scientists is that every time we make some progress and we think, okay, we understand this, then it appears to be way more complex. But thinking about it, what is exciting about the current time is that cells and organisms and organs themselves are not sitting in isolation. So the way we used to study them in the past, where we would look at one cell or one organ and say, we're going to break this apart in a linear manner, is no longer relevant. And that's why we talk of big data and we try to bring everything together. So this is, I want you to see this as a part of a work in progress, where we know a lot more now than we did 4 years ago. We have a lot of data that we're still going through. But I do see a lot. I do feel very positive about our understanding of this disease, way more than I did when I started working on this 10 years ago.
Yes, I agree. Thinking back to the very first talks that were given all those years ago, there's been tremendous progress, and Evi, you've been at the forefront of this, certainly in the UK, and contributing to European projects as well. How would this? I think you mentioned a little bit earlier on about obviously you talked about the expression of certain things being more in ARPKD and either greater or lesser than. How would that then translate into a therapy or a treatment? I mean, what do you see happening next to normalize that expression in the protein?
So the first thing we want to do, and that's what we're currently applying for, is what I showed you is information that comes from kidneys when they're already taken from the children. So they have lost function. Obviously, we want this to have value to the patient. So by the time that kidneys are taken out, that value is out of the window. What we want to study now is whether, but we all produce urine, right? Whether our kidneys are functioning normally or not. So what we want to see now is if we were to take urine from children at various points, ARPKD children, and normal, that's inspirational, but I'll say it, and normal children, so we match them. Those changes that we see in the kidney when it has lost function, can we detect them in urine prior to a situation where there's 100% no function? And if we can detect them in urine, do we have levels? So, you know, I don't go, well, we all go to have regular blood tests, blood and urine, I should say. And we have indicators for our cholesterol, for our what else we get tested for? I'll stick to cholesterol because that's an easy example. And then we can say the levels are this, the normal range is that, and we are within the range, so we're fine, or we're outside the range and we need an intervention. So I'm hoping that once we test this in urine and blood, and we find those particular proteins to also be affected there, that we can use them to distinguish between a child who has ARPKD and their kidney function is fine, and a child who has ARPKD and their kidney function is rapidly declining, so we can detect it before there's a need for an operation. So not therapeutic in the sense that we give them something and that stops the disease or slows down the disease, but diagnostic in the sense that we detect that there's a need for an intervention now, or there's going to be a need in the next month, and we intervene now to slow down disease progression.
All right, so it's part of the care and the management of children.
At this point, yes.
Yeah,
Whether,
yeah.
obviously, if we see that those changes are present, so if those levels of expression are different based on the severity of, let's take the GFR that these kids have, and we can make an association and there's some correlation, then we can look at therapies as well. But until we have 100% shown the link, it's a bit early for me to be speaking of therapies right now. I don't want to give false impressions.
Thank you. A couple of people have had to leave. Those left on the call, any questions you'd like to ask Evi? If it's easy to do it in the chat, just do it that way.
Can I ask a question?
Sure.
Oh, sorry. Go ahead.
Yeah. Hello, my name is Susanne and I come from close to Hamburg in Germany. I'm the grandmother of an ARPKD girl, 6 years old. And we have problems. She has problems now with the liver. So it's not the kidneys which start not functioning properly. They are not in a good condition, but they don't cause the problem. It's the liver fibrose and liver cirrhosis, which our PKD started with. What does it mean? Is it good? Definitely not a good, but how bad is it that it starts with the liver beside of the damage of the kidneys?
I don't think that you can call it good or bad. It doesn't mean anything. And actually, I'll give, so when that was one of the questions we wanted to ask. If I go back, as I'm going forward, I think. So one of the questions we wanted to ask when we looked at the changes on DNA was, sorry, this went a bit too far, was whether these changes were unique to individuals that had mainly problems with their kidney, or was it also present in individuals that had problems only with the liver or both with the kidney and liver? The answer is there are, and this is not just our work, other studies have done it as well. So there are children that have related children, so siblings that have changes in exactly the same bit of DNA on PKHD1, and one of them has problems with the kidney and the other has problems with the liver. So I cannot at this point right now make a prediction on whether it's going to be better or worse, because it's not the change itself. That links to what I said before. It doesn't appear to be right now with the data we have that it's the change itself that is responsible for the outcome. It's also the environment. So what other genes these children have? Do they have any that have protective functions? Do they have any that have that give rise to more severe outcomes? And what we're trying to do right now is to collect in collaboration with Birmingham Children's Hospital, they have a cohort of children that have liver manifestations, either firstly, as the case you're describing, as your grandchild, or mainly, and we're trying to see how is it different changes in the DNA that they have. So could we say that if a child is given a genetic diagnosis, and I'm guessing your grandchild has a genetic diagnosis,
Yes, yes.
and they have particular mutations, then it's normal to expect the liver to malfunction first, or the kidney, or there's no clear link? But I don't think that you should worry more or less.
Mm-hmm. Thank you very much. Thank you for your answer. May I add some silly question? Does food or diets mean anything to prolong the process a bit? Does it help?
Tess, do you want to so from a scientific perspective, I'm going to say that any dietary intervention, so if I were to be eating, I'm not a patient, but obviously if I'm eating better than worse, if I'm having my vegetables and I'm not feeling myself, I'll be in better condition. In terms of whether changing the diet is going to slow down how quickly the liver loses function or how quickly the kidney loses function, I don't think that anyone has proven this 100%. So I don't think so. People are going to tell you about salt intake, monitoring blood pressure, and that's fine, and you should be doing this. I don't have myself any evidence that it's gonna prolong kidney or liver function by, I don't know, 6 months or a year, because our study is based on genetics, and it is a genetic disease.
Yes, Evi, you're correct. There is no evidence that dietary treatment or different types of foods or different diets can help. As Evi said, because blood pressure is always involved in ARPKD. I mean, managing your blood pressure is really important, and salt plays a role. Sodium, to be precise, plays a role in blood pressure. So that's so doctors will advise healthy diets, balanced diets with protein and carbohydrates and fruit and veg, as for the normal population. But there may, of course, be children who require additional nutritional supplements because they are often, some of the children will require liquid feeding from an early age, and will require extra support. I don't know whether that happened with your granddaughter, but that's known in ARPKD, that children require specific nutritional foods or drinks, but those are prescribed by doctors and dietitians according to each child.
So I understand it's more a matter of supporting the whole body with a healthy.
That's right. Yeah. Thank you.
Thank you very much.
Daisy, you had a question?
No question, but I want to say thank you. I took part last year in Birmingham to the ARPKD day, and I learned a lot from that day, and I've been shown slides like this, and nothing. I just wanted to say thank you, and also for all your work, obviously, and very appreciated. And I know you don't want to give us false idea at the moment, but I'm not sure if I got it right, is that there should be like not a cure, but a way how to, because what I got is my grandson is taking medication and it's like to slow down the decline of the function disease. So do you think you are going to get like a better, how to say, treatment or something like that? I know there will not be a treatment because at the moment there is only the transplants sorry, I know there will not be a cure, but do you think you're working on a better treatment, or with the time we could finally actually find like a cure or something?
So very nice to see you again, Daisy, and thank you for your kind words. It was great to meet you in Birmingham, and I think you are all more, well, you are the stars, not me. We just do the research. You have to deal with the situation day in and day out. In terms of the treatment, I think what I've heard from many conversations since I've been involved with the charity is people asking, and it's a real worry, can you predict progression? Because you might have a kid whose kidneys are working absolutely fine one day and not the next. And for me, this is what this project is trying to do, to give us signals that could predict progression. So if there was a routine in the future, right? If there was a routine test that a kid could do every 6 months, and it would say how quickly it was declining in kidney function, then that is affecting the treatment pathway, whether by saying that he or she needs a transplant, we need a transplant in the next year, so let's put them up on a transplant list, or by saying that the kidneys are absolutely fine, so carry on as before, or by changing the medication that they're taking. This is how I see us as having a more direct impact. I don't think I said that I don't believe that this could have potential for a cure, because look at tolvaptan. Tolvaptan was discovered with similar studies many years ago, and it's not an ideal drug, but it has value for certain patients with all the drawbacks that it might have. What I didn't want to say is that there's going to be a cure, like we're going to prove everything I've said, and there's going to be a cure in the next 10 years. Cures can also be genetic. So there's gene therapies for many diseases right now. And this comes in from a genetics perspective. So that could be an approach, but this would mean that a specific gene alone could slow down disease or make kidneys normal again. And so far, we haven't found this one gene, because if you look at, so this is an example where we look at what happens in ARPKD kidneys. We identified a few genes that are different, not just one. And this is where our challenge lies. And this is where then I said we have to look at pathways, because when you have 10 genes that are different and you only tackle one, what is going to happen with the other 9? Could they perhaps, so whatever intervention you make, could they perhaps bring it back to the initial position or not have any effect? These are studies we have to perform before we can talk of cure. And this is where preclinical models such as animals come in very handy, because some of these studies you can do and you can see the effect without having to wait 10 years because you have a controlled environment. So I don't know if I've answered your question.
Yes, thank you. Yeah. Yes, thank you.
Thank you all. Morven and Laura have left, and thank you, Evi, for the presentation. Does anybody have any other questions, or shall we call an end? And I'll stop the recording.
If you can stop the recording, then I have a question for everyone.
Okay.
It's going to take us 5 minutes.
All right. Thank you very much, Evi, for your excellent presentation. And we look forward to hearing more about your research projects in future.